Dear learners, welcome to the program on targeting DNA structure. This will be discussed in two parts. Part 1, targeting DNA structure, deals with salient features of double-stranded DNA, various unusual forms like triplex and quadruplex, and the significance of telomere in cell division and cell death. Second part, targeting DNA structure, a therapeutic approach, deals with various experiments. So, let us start with DNA structure.
DNA is full from is deoxyribonucleic acid, DNA. There is another type of nucleic acid we call ribonucleic acid. Again, I will give little bit structural aspect of that. These are double helix. Double helix means you have seen the spiral staircase. It looks exactly like that, but there is a directionality. So, basically they code for genetic information. What I mean genetic information, there are some information which we get from our parents and some information is passed from generation to generation. So, DNA is the key part of the body which basically contain this kind of information which has to be passed from one generation to another generation. To give an idea of dimension, how much it is, how big it is basically very teeny, small thread kind of stuff. The diameter is 20 angstrom, and there are alphabets like A, B, C, whatever you did. There are some alphabets, chemical alphabets, which basically dictate the information of heredity within the DNA, and there are 4 type of alphabets, and total number of letters in human genome is 3 into 10 to the power 9 base pairs. Base pair means 2 alphabets we call one pair. I will explain again why we need that kind of pair, but that many letters are there to write all the genomic information which has to be passed through in the next generation. So, what is the scale? If you think any chemical bond like water, the structure is H2O. There is a chemical bond between hydrogen and oxygen. Typically, this length of a bond is 1 angstrom. Then come to the amino acid. What is amino acid? Amino acid is nothing but the building block to constitute a protein, like a brick of a building. So, you have different type of amino acids and that comes together, get reacted and give you protein. I think you must know some proteins we need for our day to day life. So, that building block length is 10 angstrom. By this way, the dimension of biological molecules is increasing. So, globular protein, one standard protein, it is another order or more in dimension, which is 100 angstrom. So, in the right hand side, I am giving in the same scale in the meter, minus 10 to the power 10 meter to chromosome DNA. Whatever the molecules I am talking about, they are inside with our body. There is a known as cell. Inside of a cell, there is a nucleus where this DNA stays in terms of the chromosome. The scale of a chromosome is 10 centimeter. Now, you understand this 10 centimeter of chromosome has to be within 1 micrometer or 10 to the minus 6 meter in dimension. So, there is a huge amount of condensation is happening to store this whole 10 centimeter of DNA length within this small size. So, it is a closely packed. So, what are the bases? Again little bit chemistry. There are 3 parts. One part is purine and pyrimidine. These are heterocycle which basically bond with another type of base and generate this kind of genetic code. Like say if I write ABCD like that, you will not understand anything out of it. But if I write a word WORD, the placement of letter one after that gives a meaning. So, in the similar way, these bases are arranged such a way that each and every arrangement has some meaning. Now, anybody cannot read English, so you have to train them and you have to give the meaning of each and every word. Similar way, we have to learn how to read these bases to understand what has been written. That's called human genomics. So we read base by base by base, then there is a logic to understand what is the meaning if base A, B, C is coming together, if there is a gap and so on. So by that way we are try to understand the genomic information which is encoded within our body in DNA like that. So it has three parts. One is a purine, pyrimidine, then there is a part which is basically sugar molecule, ribose, and there is a inorganic part which is phosphate. These bases what I am showing, they basically interact with the counterpart. Then the phosphate which I was talking, they basically connect to each other to create the length of the DNA or the extent of the DNA. Basically to repeat A, B, C, we need a like when we are writing like a Hindi script, we need a line top of the word. This is nothing but the line, okay, to keep them aligned. So phosphate group basically they are stitching to each other bases. So in between there is a sugar molecule, ribose molecule. Now there are two types of ribose. In one case I will have one molecule here, OH. So this is ribonucleic acid, ribose molecule. If I remove OH, that is deoxy. So in case of DNA, this OH is absent. So as I told you, these bases basically recognize the opposite base. We have four alphabet: guanine, cytosine, adenine, and thymine. These whole four bases basically dictate the novel which is written inside our body within this only four, using only these four bases. Now arranging this base gives different kind of information. Now G basically recognize C with forming the hydrogen bond. Hydrogen bond basically is a chemical bond. It is a non-covalent bond. It's very low cost effective. In all the time, what nature does basically, they create or they uses bondage which is very easy to break and easy to create, so that you don't need to spend huge amount of energy either to create something or destroy something. Now this creating and destroying, you need always release some energy or absorbing that energy, and by that way biology generates signal which basically recognized by other system that yes, there is a signal, I have to be functional right now. So this hydrogen bond basically is holding together two strands of DNA. As I told you, DNA is a double helix, and one after one base is complementary to each other. G is complementary to C, A is complementary to T, and they are both forming double helix DNA. So this is more chemically shown. So if you go back 12th standard chemistry knowledge, the hydrogen, if it is attached to a electronegative element, it has a tendency to form hydrogen bond with another electronegative element. So using that principle, all the bases within DNA is getting interacting with the counterpart of the other part of the DNA. So again, this is another understanding. So I have told that how single stranded DNA is built up. There is a unit, one unit interact with the second unit, like that it can grow, make a polymer. Now they interact with the counterpart, all the opposite strand, give this kind of double helix, and this is the chemical representation of the double helix. Now once these are extended, as I shown here, they get coiled and coiled. There are proteins which basically helps them to be coiled. Histone, these are very positively charged protein, and DNA is the negative charge. Then they get condensed. Once condensed, basically form the chromosome. This chromosome goes within the nucleus of a cell. All the cells in your body, there is a nucleus, and nucleus contains the chromosome, and chromosome is made of double helix DNA which is condensed in the nature. Now here I want to mention the end of the chromosome is no longer double helix, it is a single helix. Okay, we do not have at the end double helix, there is a only single stranded. I will come back why I am mentioning, why I am trying to point out another feature here, that I want to use this information to design my research problem. If we grow, new cell has to be created and some old cell has to be die. That is why we, from baby to become adult, then adult to go become old, and during all this process always new cells are forming. One cell is getting divided, giving 2 cells, again 2 cells is getting divided, 4 cells, and so on. Now, when in a certain case number of cells is not required that many, then cell try to die. It is happening. We are not keeping all the cell whichever we have in at the baby stage to the development. New cells are forming and some cells are going to dead. Now, how cell decide which cell has to be die? This is a representation of chromosome, and the end of the chromosome, as I told you, single stranded. Okay. Every time a cell is getting divided, that length of the single strand is decreasing. Every time I have shown say 4 steps, always it is decreasing to end of the chromosome, which is known as a telomere. This is the process when it is getting divided. There is another protein machinery which try to extend the end of the chromosome or telomere. So, there is a balance. One process is trying to extend, another process, cell division, is trying to decrease. Now, there is a competition. If the division is becoming more prominent and once a length is becoming a critical length, then cell gets signal that I have to die now. I cannot replicate further, I cannot divide further. So, length of this chromosome will decide whether a cell is going to die or is going to divide further. Now, in a stage which is basically not normal stage, this protein machinery somehow is becoming overactive. That means it is extending very high efficiently. As a result, your cell is getting replicated, but the length of this end is not decreasing. So, as a result, cell is not able to decide when to die, and cell become immortal, and that is the cancer state. Cancer is nothing but the unprogrammed well cell division. It is a growth of cell basically, and now biology cannot control when to die, how to die, and cell become cancerous. That is the way cancer happens most of the cases. Now, as far as I have explained, we know Watson-Crick base pair or this 4 base pair, as I telling you, we know so many information that today I can write a program in a computer and design small nucleic acid structure and come out different kind of structure which I have shown in my slide. You can come with a bulky, like a ball, like a box, all this kind of structure you can make very small teeny size like a nanometer scale with using nucleic acid. But in nature, as I told you, most of the cases is double helix, but it does not happen in that way. Sometimes some deviation happens. That means there could be different kind of structure than the double helix which I was explaining. So, what are those structures? They could be another strand like 2 strands, another further strand, 3 strands come together can make a structure. These are triple helix, but it is not in a normal case. Generally it does not happen, but it may happen in certain cases. 4 strands come together and can form a structure. Again, it is not a usual structure which happens generally within the DNA, but it is an unusual structure. It may happen in certain conditions, could be a disease condition. So, my research focus on to understand this kind of unusual structure, because I know in a normal case DNA is a normal double helix. Now, if I see that there is a disease and there is an unusual structure formation, I will correlate whether I can use the information of unusual structure to prevent the disease. Either I will stop to form this unusual structure, or I will block the structure such a way that whether structure is there or there is no structure, there is no difference. So, it cannot execute the disease state. That is the basically major focus of my research. Today, I am going to talk about a special structure which is 4-stranded DNA structure known as a quadruplex. So, what is the quadruplex? There are 4 bases, as I told you: guanine, cytosine, they base pair each other. There is another base pair, adenine and thymine, they also pair. But guanine is a one type of base pair, they can come close to each other in presence of monovalent cation like potassium or sodium, as I showed you in the square box region. They can form again hydrogen bond among themselves and create a kind of network, that you need at least 4 guanine together in a same plane. Now, if DNA there is a situation this 4 guanine can come close together and there is a monovalent cation, this kind of 4-handed structure can form, which is known as a quadruplex. So, what is my primary requirement? I have to have guanine conjugatively for few stages, at least 4 stages in the DNA, so that they can come closer and give you 4-stranded DNA structure, which is known as a quadruplex. Again, it is not a usual structure, it is unusual structure. It forms in body in several places. We have done extensive research, try to understand computationally where there is a probability to form this kind of structure. Then we have taken this computationally predicted structure to the test tube to understand really they form or not. That is a physical chemistry we do. One, if I see that yes, there are certain structure it forms, then I try to understand in cell system whether this structure have played any role or not. Now, if it is playing a role, whether it is essential role or abnormal role, if it is a abnormal role, I try to rectify with some chemical engineering molecules or chemical engineer nucleic acid. That is the theme of my research. Now, coming back to my earlier slide, as I told you that end of the chromosome is a single strand. Now, if you try to read the letter by letter of this end of the chromosome, you will see that you get GGG, AAT, GGG, AAT kind of repeat. Okay. What I told to form a quadruplet, what is the essential? That you need few stretches of G. Now, again if you remember this slide, this protein machinery, what they are doing? They are extending the end of the DNA. How they are extending? The protein is coming and recognizing the single stranded structure, first binding, then doing some synthesis and extending. Now, if I have a G rich region and if I somehow stimulate or can give some condition, this single stranded structure is not any more single stranded structure. It can form some different kind of structure. In that case, this protein machinery will not be able to bind to the single stranded structure and cannot extend the length of the telomere end. So, if it cannot extend, that means cell has to die. Now, in cancer, as I told you, it is overactive. So, if I have a molecule which can go and bind to the single stranded structure and create 4-stranded structure, that means this protein machinery will not be able to bind anymore and extension cannot happen, which means that if I can demonstrate it, that I may have one way to tackle cancer. Here I will stop, thanking once more to all of you.